Thank you, Kata, and thank you very much for that introduction. So when Kata and the other organisers asked me to present a talk on pseudoscorpions, I was given free rein and you know, asked to come up with a title and a content that suited me best. So I want to talk about how molecular data have transformed our understanding of pseudoscorpion phylogeny, but I always like a little hook at the beginning of my talks, as a lot of us do. And so I started by, the beginning of the title says from trichobothria to genes, and I'll explain why in a second. Now, first of all, as any of you who know me, I struggle at English, especially at 1 AM in the morning, but I have no other languages. And so this, apart from ordering a beer, is the limit of my Spanish. So we'll conduct the rest of it in English, if you don't mind. Now, pseudoscorpions are a cosmopolitan order of arachnids. They're found essentially in all terrestrial habitats, except the polar regions. You do get them right down to the intertidal zone, so you can get them on beaches in some parts of the world, and they spin a little silken chamber that they cover themselves up in when the tide comes in, and they occur all the way up to the highest slopes in the world, including on the slopes of Mount Everest in the Himalayas. This is a distribution map of all the type localities of pseudoscorpions that have been described around the world. So you can see that not only are they on all of the continents, but also they're on many oceanic islands as well. So some of them can disperse to quite far-flung places. So these are only type localities, obviously, and more widely distributed than that, but they're truly a cosmopolitan order. They come in all sorts of different shapes and sizes. They come in essentially one colour. They are brown. They can be light brown, dark brown, yellow brown, black brown, red brown, but mostly just brown. Some of them do have cute colours. They have little spots on them occasionally, especially some of them on the carapace and on the tergites, but essentially they come in one colour. They're also in size. They range from, the adults of some species are 0.7, 0.8 millimetres in length, the body length from the front of the carapace to the tip of the abdomen, all the way up to big ones here. This is the genus Garypus on the right-hand side. There's a species from Ascension Island in the Atlantic Ocean called Garypus titanius that the body length is about 1 centimetre long. So they're about the biggest there are. Quite common if you know how to find pseudoscorpions. They're not difficult to find. You just got to keep your eye out for them. There are currently 26 recognised families in the Holocene fauna, about 460, 70 genera, 3,700 valid species recognised at the moment. Now, this is the slide that links the rest of the talk to the title that I was talking about. And when I say from trichobothria to genes, it's remarkable that the trichobothrial position of, or the positions of the trichobothria on the chela of the pedipalps, which of the chelar fingers here, all pseudoscorpions have got these chelate pedipalps like scorpions. They have often a set number of trichobothria. There are 8 trichobothria on the fixed finger of the chela and 4 on the movable finger. In the 3 other post-embryonic instars, which are termed the protonymph, there's a typo, the deutonymph and the tritonymph, they have 3 and 1. The deutonymphs have 6 and 2, tritonymph 7 and 3, and the adults 8 4. So sequentially, these trichobothria are added at each of the molts of the post-embryonic instars. And not only is it important in determining the nymphal stages, but also the positions of the trichobothria vary between the genera, and sometimes the species. So they're quite important in pseudoscorpion taxonomy. And that's the last time I'm going to mention trichobothria for the next 15 minutes. I'm going to move on to phylogeny. So I guess the first phylogenetic tree, or as this person called it, a scheme of classification, published was by Joseph Chamberlin in his magnus opus of 1931, which was essentially his PhD thesis. And he produced this tree, which was a tree-like representation of his classification, which was very, very influential, that he developed in 1931, and which shows all of the families at the top here, the superfamilies and the suborders of the Pseudoscorpions that he recognized at the time. Now, this classification was essentially in place for the next 50 years, 60 years, and it was an intuitive classification, so it wasn't based on any analysis at all, which you would imagine in 1931 wasn't done. The next time there was any investigation into the phylogeny of Pseudoscorpions was a paper I published in 1992 on the phylogeny and classification of Pseudoscorpions. And for those of you who are old enough to remember Hennig 86 in the pre-molecular days, I was able to run a phylogenetic analysis based on 126 characters. At the time, it was palatable to be able to score families for characters and not species level phylogenies, but we recognized 24 families at the time, and I produced an analysis that was published. Turn my alarm off. And this is the resulting tree on the left-hand side. This is part of it you can see here. This clade here runs to this part of the tree on this side. And for those of you also familiar with Hennig 86, outputs included each of the synapomorphies or autapomorphies for each of the family level taxa at the top here are listed on the phylogeny. I'll come back to this in a slightly different form later on, but you'll see that some of the families, some of the clades were very poorly supported when it comes to characters that could be used to define each of the clades or even some of the families. The next paper that dealt with phylogeny in the broadest sense was published by Jérôme, Murienne, Gonzalo Giribet, and myself in 2008. And we published a 3-gene phylogeny based on a number of different species of most of the Pseudoscorpion families. This is the resulting tree on the left-hand side. I'll summarize it in a second, but you can see the colors define each of the major groupings, the superfamilies. Here, there's Feaelloids at the top, Chthonioids. The thing I want to draw to your attention is some of the anomalous positions. There was an example here of a superfamily where one taxon ended up somewhere else. I won't go into detail, it's not really relevant, although in hindsight, this turned these two issues that we have here. This one was a contamination issue, and this one was sort of something that we resolved later on, which I'll talk about shortly. But in general, the superfamilies were moderately well supported, and it helped outline the phylogeny of the group at the time. The most recent study was last year, and we published a paper with Ligia Benavides, Julia Cosgrove, Gonzalo again, and myself. And we accumulated 41 Pseudoscorpion species and their transcriptomes, which included 22 families that were currently recognized. So we had all of the families that were currently recognized except these 4 representatives here. So it was pretty good coverage. I won't go into the analytical details. Suffice to say that for the final analysis, we were able to obtain over 200 genes for the analysis that I'm about to show you here. So that's a vast improvement on the 3 markers that we had in the 2008 paper, and certainly a hell of a lot better than the Hennig 86 analysis that I ran in 92. This is the resulting phylogeny. I'm not going to go into a huge amount of detail, except luckily the Pseudoscorpions came out as monophyletic. Yay, good for us. We also, you can see by these little Navajo charts here that each of the different forms of analysis and their support values are colored here. Most of them are pretty good, 100% support, with occasional lower levels for some clades in different analytical methodologies. But in general, it was a pretty well supported tree. And I'll talk you through some of the outcomes from that now. For those of you who are interested, one of the graphics that we presented in the paper was this, which has, I'm a bit of a nerd when it comes to previous classifications, and we were able to look at all the past classifications and the different changes that the families had made over the years with the different schemes and phylogenies that have been published. So they're all summarised there. Now, some of the major changes are that the suborder Epiocurata is not monophyletic. So let me talk you through that. My analysis in 92, these two superfamilies, the Chthonioidea and the Feaelloidea, were grouped as a monophyletic group, which I termed in the suborder Epiocurata, which means gentle hand. The rest of the families were the Eocurata, which means poison hand. And by that, it means that this group of pseudoscorpions in the blue are all of those that have got a venom apparatus in the keel or fingers, which they use to subdue their prey and defend themselves. These two superfamilies here don't have any venom apparatus. And because of the monophily, the perceived monophily of these two superfamilies, I put them in their own suborder. The paper we published, the 3-gene paper we published with Jérôme, didn't recover that topology other than the Eocurata was monophyletic and a little bit of a change with the superfamily structure. The Epiocurata was paraphyletic with the Feaelloids, sister to the rest of the pseudoscorpions, then the Chthonioids. The transcriptome analysis we published last year recovered essentially the same topology, except that the Chthonioids and the Feaelloids were switched in their position. But once again, Eocurata was found to be monophyletic. What we also found was that the outcome from that is that in the paper last year, we proposed a different subordinal structure. We left Eocurata as one of the suborders. The Heterosphyronida, which includes the Chthonioids, which was a name that Chamberlin proposed in 1931, was retained in its own suborder. And to reflect the unusual position of the Feaelloids and their morphology, we placed them in a new suborder called the Diplosphyronida, which just means strange legs, which they do have very unusual legs for pseudoscorpions. So that's one of the biggest changes we made. As I mentioned, this suborder Eocurata have got venom in their chelar fingers. This is a chelar of some pseudoscorpions with the venom duct shown off to one side. And in 2008, there was a transcriptome analysis that we published with some colleagues looking at pseudoscorpion venom, if you're interested in some of the details of what we're able to find of the enzymes and protease inhibitors that are in pseudoscorpion venom, which was quite interesting. Some of the other changes included two of the families that were recognised previously, Chthonioidea and the Lectiteidae, were heavily embedded, and also with other studies that I've been doing on morphology, embedded within the Chthonioidea. So we just made them into a single family and synonymized those families. And a subfamily that was previously recognized as part of the Olpiidae was elevated to family level, based not so much on the transcriptome evidence, but other data that we had available. So we just formalized that change. One of the strangest things that occurred was that the superfamily Garypinidae was not found to be monophyletic. You can see here I've highlighted this part of the transcriptome tree. All of the representatives of Garypoids, including Garypus, the Garypidae, the type family of the superfamily, were here. And the other two families, the Garypinidae and the Larcidae, are actually sister to this bigger clade down here, rather than being sister to the Garypoidea, the rest of the Garypoidea. This was, to me, really unusual because of the morphologically, they were not distinguishable from each other. But my 1992 analysis found that the Garypoids were very, very hard to define and to find autapomorphies for, synapomorphies for. So it wasn't all that surprising. And yet, even in the 3-gene analysis, which was run by Jérôme and Gonzalo, we found that here are the classic Garypoids, and these are the Garypinoids up here. We didn't recover the Garypoidea as monophyletic in this analysis either, notwithstanding the issues of the this contamination here, or the Geogarypids up here, which is just probably a lack of gene coverage. So it was hinted at as early as that 3-gene study. So we were quite happy to formalise that change as well. I just want to talk quickly about the age of Pseudoscorpions. It appears that all of the families had appeared by the Mesozoic. In fact, all of the Mesozoic fossils that have been studied so far, and there's been quite a number of studies now. I've been looking along with other colleagues at specimens, especially from the Burmese amber deposits. They can all be attributed to modern families. So we've yet to see anything that stands out as unusual and doesn't look like a modern Pseudoscorpion. In fact, some of them I've looked at so far, including, I won't go into details, but a couple of species look as though they are represented in Burmese amber, which is 100 million years old, and yet can be attributed to modern genera, which is, I think, pretty unusual. So it shows a huge amount of morphological stasis in Pseudoscorpions over a phenomenal period of time. This is the sort of a modified phylogeny taken from the transcriptome analysis of Pseudoscorpions, stripped down so you can see the families on the right-hand side here. These are all the superfamilies that are recovered. And this is the dating analysis we were able to do. And it was underscored by fossil taxa, with the oldest being these Cretaceous fossils occurring with the crosses against the Burmese and Bitterfeld ambers down the middle here. So you can see that there's really nothing that's inhibiting, as I said before, the fact that all of these families are Mesozoic in origin and often much, much older. So it looks as though many of the major clades developed in the Paleozoic. So yet again, one of these really super old arachnid orders. I just lastly want to finish on my title talks about from trichobothria to genes. This is just a teaser for a study I'm conducting at the moment. This is a CO1 phylogeny only, but it is a molecular phylogeny. You'll all be tested on this next time I see you in person, so take notice. Got it? Got it? Got it? Okay, there it goes. So this is a genus of Pseudoscorpions that occurs only in northwestern Australia. This is the Pilbara of northwestern Australia. They also occur on Madagascar and India. The sister genus is Haya, which is in southeastern Asia, which we have a Burmese amber fossil, and that's 100 million years old. And I can barely distinguish the fossil from the two recent species, the Holocene species that occur there today. We found, partly using sequence data, but also using morphology, 20 new species of this genus in the Pilbara. There are 3 on the surface, 17 that only occur in the subterranean environment. And the reason I've shown you this is that we're going to go ahead and publish this paper, and we are also going to describe the 7 species for which we only have nymphs based on the CO1 data at all, because the morphology is not clear with nymphs. You really need adult specimens to do Pseudoscorpion taxonomy. But a lot of these species occur in highly threatened landscapes because of mining activities that occur in the Pilbara. For any of you familiar with how the Australian economy operates, the Western Australian economy is all on iron ore that we sell to China. So all of these areas are mining deposits. So we'll go ahead and formally name all of those species using morphology, but in the case of the species only with nymphal specimens using the CO1 data. So with that, I'd like to thank you very, very much for listening. Thank you very much for inviting me. It's so exciting to participate with my colleagues and friends in other parts of the world. And I'm happy to answer any questions.